Hi everyone, so this recording is going to go over the Properties of Water lab. Take a couple of minutes to make sure that before tomorrow you read the introduction. I'm going to talk a little bit about polarity. Polarity is going to be caused by which element in the molecule is kind of holding on to those electrons more than the other element. So water is made up of hydrogen and oxygen, and the oxygen atom is the larger one in this image. And the two hydrogen atoms are a little bit smaller and they have less protons. Because of the fact that the oxygen atom is larger and has more protons, those protons having a positive charge are really going to pull on those electrons. So if you stop and think about that for a minute, you could probably figure out what kind of charge the large oxygen atom is going to have if they're kind of pulling those electrons a little bit more towards him. Those electrons, remember, have a negative charge. So think about that when you're discussing this page with your team. You can go ahead and measure out 10 grams of NaCl, which is salt, and you'll be measuring out about 100 milliliters of water, and you want to identify with your team what the solute is and the solvent is. Now, during class time, if you need to, you can look up those two words in order to determine the answer to that. On part 2 for the lab, you are looking at hydrogen bonding, and you need to label the 4 hydrogen bonds in the diagram shown. And then again, if you need to work together to look up and find out what a hydrogen bond is, what an ionic bond is, and a covalent bond is. And then below there is a spot for you to describe a test you could do at home to prove that water expands as it freezes. You do want to give a complete explanation there when you are working on this with your team. On part 3, this is the first part that you're going to be doing an actual experiment. So notice in the procedure it says obtain 2 pennies, 2 pipettes, a beaker of tap water, and a paper towel. And then you'll also need a beaker of salt water. Those will be at your lab table. There'll be some salt for you to make some salt water. Read the instructions. It says you're going to be using the pipette to drop tap water onto the penny 1 drop at a time and count how many drops you add before it spills over the edge. Record that in your data table. Notice that there are several trials for you to do and then take an average. And then there are 2 questions for you to answer underneath there. For part 5 in class, you do not need to have everyone make a graph. There is 1 graph paper at your lab table, and you can put someone in charge of graphing the points and then someone else in charge of reading off those points so that you can make the graph pretty quickly, and then everyone can take a picture of it and enter that picture into the lab here. And then you've got some questions to answer. And then part 7 has you labeling the meniscus on the graduated cylinder. And then part 8, instead of using celery, we're actually going to use capillary tubes. So capillary tubes are very tiny little tubes where the water will go up it, just like they would in the little tubes that make up a celery stalk. And so you can make observations immediately. You will not have to wait a certain amount of time. So just record what you see happening to the liquid in terms of what happens when you place the capillary tube into the liquid. And then you have some questions to answer on that part.